Hello everyone, today we'll talk about the process of DNA replication. So let's start with it. The very first step of DNA replication is initiation. In this, what happens? There is unwinding of DNA by the enzyme called DNA helicase. Unwinding of DNA by the enzyme DNA helicase. Right? So now these unwinded DNA, these two separated strands of DNA which we have separated by the enzyme DNA helicase, these two strands are stabilized by certain proteins which are called single stranded binding proteins, SSBP. These stand for single stranded binding proteins. These proteins stabilize the DNA strand so that they won't get wind again and make the helical structure. So these helps in the making of the replication fork. Replication fork, which is Y-shaped, as you can see in the figure. Right? So now let's talk about the second step of DNA replication. This process is called elongation. Right? So in this, what happens? The separated strands of DNA, the Y-shaped separated replication fork. So in this, there is an enzyme which we add, primase. It initiates the DNA replication. It initiates DNA replication. How it does that? So at the 3' end of the replication fork, so it adds certain 10 to 60 nucleotides. This particular long nucleotides are called primer. So a primer is being added. This provides the initiation point for DNA polymerase enzyme to polymerize this activity, to add more nucleotides with the help of deoxynucleotide triphosphates that we studied in the enzyme sections. We have 4 types of DNTPs: DATPs, DTPs, DCTPs and DGTPs. Right? So these 4 will add the 4 bases of the DNA strands. Right? Now let's talk about this in more detail. So replication of DNA in this direction, in 3' to 5' direction, is continuous. It is continuous, and replication of DNA in 5' to 3' end is discontinuous. Now how is that? Let me talk about it in more detail. So the formation of DNA strand in this direction, in 5' to 3' direction, is continuous, means there is a continuous strand of DNA. However, in the other direction, which is the 5' to 3' end, this direction, there will be a formation of DNA strands like this. So this strand is called, this red strand is called continuous strand, and these strands which are discontinuous, we call this strand as lagging strand. Now there's a reason behind it. The enzyme DNA polymerase, it only polymerizes the activity in 3' to 5' direction and not the other way around. It does not catalyze the polymerase activity in 5' to 3' end of the DNA, right? So they only make the activity continuous in this particular direction. That is the reason the other side has discontinuous strand. But what happens, these lagging strand, these broken strands of DNA are called Okazaki fragments. Okazaki fragments. These Okazaki fragments are called Okazaki because there was a scientist who discovered these strands. The name of the scientist was R. Okazaki. These fragments are later on joined by the enzyme known as DNA ligase. So these strands are joined by an enzyme called DNA ligase. Once the entire polymerization activity is complete, these two strands of DNA, this one and this one, will be released and make a helical structure of its own. I hope you understand the process of DNA replication. If you have any questions, you can leave a comment. Thank you so much for today and have a great day.